Thank you for the nice introduction, and welcome also from my side from the windy Netherlands. And yeah, I'm going to present to you the paper that we published last year, Home is where the lipids are. We compared two nanodisc systems on a membrane transporter. And I have a quick outline of the talk shown here. And I would say we start with the biological background. So the main character of this publication was a protein called GLTTK. It's a homotrimer from Pharmacoccus codacarensis. Each of the monomers is shown here in a different color, and it transports aspartate in symport with 3 sodium ions. Glutamate transporters have a highly conserved binding site, which is why we chose to work with these model proteins. But however, since GLTTK is a membrane transporter, it requires its lipid environment around it to function properly, right? So how can we study membrane proteins outside their physiological environment? And for that, we use membrane mimetic systems. In the labs that I have worked, we mainly focus on glycoamphiphils, membrane scaffold proteins, and polymers. For this publication, we focused on glycoamphiphils and membrane scaffold proteins, more specific on one glycoamphiphil and one variant of the membrane scaffold protein, but I will come to that in more detail now. So glycoamphiphils are small molecule amphiphils that form native lipid nanodiscs that contain the membrane protein. They can extract the membrane protein with its native lipids around them. Shown here are now 2 structures. So on the left, you can see DDDG, which was the glycoamphiphil that we worked with in the paper. On the right side, this is the newer generation of glycoamphiphils called the BGCs. The abbreviation is for bisglucosides. They are already tested, and they seem to even have better properties compared to what I'm showing now to DDDG. But however, the paper was with DDDG, so we stick with that for now. And we compared it with the membrane scaffold proteins, short MSPs, which are a self-assembled complex of lipids and a helical protein. And this helical protein surrounds the membrane proteins by this specific MSP belt. And MSP is derived from the sequence of apolipoprotein A1. Here we see the characteristic belt. And there exist many variants of these belt proteins. We chose to work with one, but I will come to that when I speak of the workflow. More important for now is that there is a reconstitution step always needed if you want to get your membrane protein into an MSP nanodisc. So you first need to extract, and then you need to reconstitute, but I will come to that. First, the aim of this paper now was to compare MSP and DDDG nanodiscs to assess their impact on membrane protein structure and function. Therefore, we first did extraction purification, and then we did some characterizational experiments. So how now does this extraction with native nanodiscs work? Very briefly. For GLTTK, we start with E. coli, break the cells, prepare the membrane vesicles, and then we add DDDG, incubate overnight. And DDDG starts to gently extract the membrane proteins together with the native lipids. The next day, we run an ultracentrifugation and then run a column to get the protein. Then we can investigate the protein and run a gel. That's what we usually do to check if everything is in place. Compared now to MSP, and now we see MSP 1D3D1 is the variant of the MSP that we worked with. We again start with the same system. So we have GLTTK overexpressed in E. coli, break the cells, prepare membrane vesicles, add in this case detergent, DDM in this case. So we don't get the nanodiscs right away. We get something is called like a mixed micelle. And then we reconstitute in a specific ratio of, in this case, E. coli polar lipids, because the protein was expressed in E. coli, and the membrane scaffold protein belt, in this case, MSP 1D3D1. Then we need to get rid of the detergent, of course. We do that with some kind of biobeads. Then we need to get rid of the biobeads. We run a column and we apply it on a gel. And spoiler, this works. So I have a gel for representation shown here, which is native nanodiscs. Let me just get my pointer. So the S here is for supernatant, where it's expected that there's just a lot of protein. The band for GLTTK should run around a bit below 35 kilodaltons. And it's important that it's not in the pellet, not in the flow-through, and also not in the wash fractions. But once we start to elute, in this case with imidazole, because GLTTK has a his tag, we see the band. And then we pulled it all together, the band is still there. We then concentrate it, apply it on the size exclusion, and then run our experiments. We also confirmed the band with a Western blot. And after running several purifications, we compared the yields. So starting from 30 milligrams of membrane vesicles, this is what we end up here, which is very similar for DDM and DDDG. Of course, for MSP, it's lower, but it has to be considered that you start reconstituting after you extract it, in this case with DDM, so it makes sense that the concentration is lower. Also, the reconstitution efficiency for membrane scaffold protein is never 100%. It varies from protein to protein, but for us, it was around 50-ish. But for MSP, we were 100% sure that we have the lipids already in the membrane, because in the nanodisc, because we reconstitute them, right? So for DDDG, we wanted to confirm that we have the lipids there. So we did mass spectrometry to determine the lipid composition. And now shown here, in the inner circles, is always the GLTTK nanodisc, the DDDG nanodisc where GLTTK is embedded, and the lipid composition of it. In the outer membrane, this is the E. coli membrane without any nanodisc, so just the membrane. And PG, for those who don't know, is short for phosphatidylglycerol. PE is phosphatidylethanolamine, CL cardiolipin, acyl PG, acyl phosphatidylglycerol. The first number is the number of C atoms, and the second number is the number of double bonds in this case. And these are all lipids that are abundant in E. coli. And I think what we can see quite well is that DDDG extracted exactly the E. coli membrane lipids, except for here, where it's a bit different, but acyl PG compared to the other three is least abundant. So yeah. So now we have two nanodisc systems where our membrane transporter is there. And now we wanted to characterize them or see if we can get any differences based on the nanodisc. So we wanted to test for thermal stability. We diluted the protein to a certain concentration, incubated it for 30 minutes from 30 to 86 degrees Celsius. And I think we can see quite well here on the gels that the band intensity is decreasing. We also quantified it below, but I think we can also see quite well here. So for DDDG, there is no protein left at 75 degrees. But for MSP, there is still a very light band left at 86 degrees. So the protein remained more stable in the MSP nanodisc, which is probably because it's a more compact nanodisc. But for this reason, we wanted to see how they look like. So we did negative stain electron microscopy, and we see DDDG nanodiscs here. So this is GLTTK in these nanodiscs, and they have a varying size from 25 plus minus 5 nanometers, which is quite on the higher side. But this is something that was expected because these native nanodiscs don't have a restriction in size. So sometimes they will extract more lipids, sometimes they will extract less lipids. That's why it's more heterogeneous. Also, the background here is not noise. It's because DDDG requires a critical mycelial concentration. And this is why it looks a bit more noisy compared to GLTTK and MSP1E3D1, where we have a more homogeneous size and also smaller nanodiscs. But this is, of course, restricted by the belt because this MSP belt should have a size of 30 nanometer-ish, and this is what we saw. And apart from that, we also wanted to test the binding. So we did a so-called uptake assay where we checked if GLTTK is able to bind to aspartate, and it remained functional in both nanodiscs. This is now shown here. So on the left side, we see the graph for MSP. On the right side, we see the graph for DDDG. And there is two curves on each graph, because if you remember from the very beginning, I said it transports aspartate together with sodium. So without sodium, it's not able to bind to its ligand, which is why we have these curves here serving as a control. But once we add sodium, we get a nice binding curve, and we could determine the KD. And the KD seemed to be 4 times lower for DDDG, which also again might be because of the nature of the nanodisc. So probably the protein is conformationally more flexible in this nanodisc. And yeah, this already brings me to my summary where I made a nice table for comparison. So very briefly speaking of workflow, it's a huge advantage because you have a direct extraction in DDDG, whereas for MSP you need to reconstitute to get your nanodisc, but you can choose the lipids, you can vary around with the lipid composition. However, for DDDG, you get the native nanodiscs, but the size is heterogeneous compared to MSP, where it's more homogeneous. So this might be an advantage when it comes to structural work, especially. And speaking of conformational flexibility, there was a higher affinity in DDDG. And this is probably, again, because MSP has this membrane scaffold protein belt around it, which might restrict the flexibility. And with that, I am done with my talk. I thank you all very much for listening, and also a huge thank you to all of the people that are listed on this slide, because without their support, this work would have not been possible. And yeah, happy to answer any questions.
Fantastic. Thank you so much, Alexandra. What a nice talk. Really well presented. We have a couple of questions in the chat. How about we ask Salma, would you like to unmute yourself and ask your question? And then we also have quite a few from Michael and Xizhang. So if you guys want to just go ahead, unmute, ask your question.
Sure. Can you hear my voice?
Yes.
Yes.
Thanks for that's a great talk. My
Thank you.
question is, what makes MSP nanodisc has lower GLTTK yield comparing to DDG, though it is more stable? It looks more stable than DDDG.
Because if you go back to the workflow, I think it's here. Yeah. For MSP, I first need to extract in detergent, so in DDM. So I am at the step where I usually have DDDG, and then I just continue working with DDDG. But for MSP, I cannot do that because I need to reconstitute the whole protein, and it will never reconstitute everything. So you need a specific ratio for lipids and membrane scaffold protein belt, but they will never reach 100%. So there is a huge loss in protein, unfortunately.
I see. Thank you.
Hello.
Yes.
Hi.
Great talk. Here I have one question about the lipid composition. So you after using DDDG, right? And you
Yes.
you just analyze the lipid composition. I guess you should have repeat multiple times. Can you see the consistency for the lipid composition from different experiments?
We only tested this for GLTTK. We also tested only the membrane, and we tested, of course, the buffers as control. For MSP, we know with which lipids we reconstitute. So in this case, this is an E. coli polar lipid mixture and MPC. But we did not run it for other proteins because it showed us clearly that it's the E. coli proteins that are extracted.
What I mean is for this protein, if you do several times, right? Then can you see the same, or can you see a big variation for the lipid composition?
No, it remains very similar because it's of course it depends if you express it in a new batch, then of course the lipids may vary a little bit, but that is anyways because it's biological replicates, right? But it tends to extract the same lipids that there are in the membrane as well.
That's nice. Yeah, I recall I used some copolymer to extract my protein, and I also did lipid composition analysis. I did 3
Yes.
times, but the compositions look quite different. So yeah, I don't know why. Yeah, but it's great to hear that you have consistent
Yeah.
lipid compositions. Great, thank you.
Thank you.
I just had a quick question about how people can access those new amphifiles that you mentioned.
Yeah, so they are produced by collaborators from the Technical University in Graz. This is a group from Tanja Wodnik, a collaborator from Sandro Keller. So the patent is from the group of Sandro Keller. But I am not sure how far this already is with if you can access them easily, but probably easiest way to just contact Sandro.
Okay, great. And can you just mention about differences in the non-specific interactions of the different amphifiles and MSPs with membrane proteins? Like, do they have different effects in terms of binding non-specifically through charge-charge interactions or aromatic interactions? I don't know. Do you see much difference there?
Yeah, so for the ones that I tried, we have not seen big differences. That's why we chose to work with DDDG in this case, because it's not charged. But I have not looked into that.
Great,
more.
thanks.
Yeah, I have one more question about the stability of your membrane protein in DDDG. Have
Yes.
you did you do any experiment to test the stability?
You mean like a differential scanning calorimetry or something like that?
You like the function at different temperature or incubation
No,
at
so
different temperature for different times.
what I do here is, yeah, of course, I first did incubations at different temperatures, but then with 30 minutes, I saw the nicest difference in how they show what they show on the gel. But they are anyways incubated for 30 minutes for each temperature, and then I applied them on the gel. I cannot do differential scanning calorimetry with this membrane protein because it costs me a lot of sample. So I would purify
I
probably
see.
a month or so for one measurement.
Okay,
I
great,
have a
thanks.
quick question, if you don't mind. You said at the beginning of your talk that you have tried polymers before, but today we're focusing on MSPs and your other amphifiles. Now, you've obviously worked with that before, so could you maybe give a little bit of insight? Did you see any key differences between this stabilization systems and the actual lipids that you're seeing, the thermal stability, and then also based on workflow, you pointed out pros and cons on both sides on the different workflows. When working with polymers, do you feel you overcome quite a few of those?
So the thing is that I worked with polymers, but I early focused on working with DDDG. So I know that, for example, the workflow is very similar with the extraction for DDDG. You have similar yields. You also have the native lipids retained. Probably also you don't have the size limitation, but that of course depends on the polymer that you use. So it's more on the side of DDDG, I would say. So I would say MSP is a different, far off group from this. But I have not tested any binding yet or something similar.
There's
I'm
a
just
question
curious.
sorry, there's a question from Nia. Feel free to unmute. Did you produce the MSP protein yourself? Were there any limitations
Yes.
during its production?
It's expressed like a soluble protein in E. coli, and it's just like a normal soluble protein with a his tag. You clean it. There is one limitation if you reconstitute, because you need to somehow find out that there are also empty MSP nanodiscs, right? And that there is also GLTTK with nanodiscs. But we found a good solution for that, and that is we cleave off the his tag after. So we purify it and we cleave off the his tag of the MSP. And once we reconstitute, we then not take the elution fractions because all of the his tag MSP is still there, but we take it from the flow through. So that's one thing you really need to consider if you work with MSPs, to see that you only have that you don't have empty ones, because you cannot see it on the size exclusion.